at 8,336 meters below the surface of the Pacific Ocean, in complete darkness and under pressure exceeding 1,000 times what a human body experiences at sea level. A remotely operated camera filmed a fish moving calmly through the water, alive, undamaged, and operating normally in conditions that every biochemical model in marine science said were physically incompatible with vertebrate life. That footage, captured by a University of Western Australia research team in 2023, did not just break a depth record, it broke the theoretical framework that established the record in the first place. This video is about what the Mariana Trench actually contains, not the version taught in classrooms, but the version that emerges when you line up the footage, the published data, and the documented history, and watch them refuse to agree with each other. The Mariana Trench is a crescent-shaped geological scar more than 2,540 kilometers long, located approximately 200 kilometers east of the Mariana Islands in the western Pacific. Its deepest confirmed point, a location called Challenger Deep, sits at 10,935 meters, or 35,876 feet below the surface. If you placed Mount Everest at the bottom of Challenger Deep, the mountain's summit would still sit more than 2,100 meters underwater. That is not a figure of speech. That is the measured geometry of the place. The first serious scientific effort to study life in the trench was Danish, not American. In 1951, the Galathea expedition lowered sampling nets more than 134 times into the hadal-zone, defined as any depth below 6,000 meters, and retrieved organisms that contradicted every prediction marine biology had produced up to that point. The Soviet Union ran its own deep trawling programs through the 1950s. Both programs found the same thing: life, in a place where the combination of complete darkness, temperatures near freezing, and pressures equivalent to the weight of 1,600 elephants per square meter should have made complex biology functionally impossible. That was the established starting point of hadal science. Almost nothing discovered since then has fit cleanly inside it. The researchers doing this work are not amateurs operating on the fringes of their field. Dr. Thomas Lindley of Newcastle University, working alongside teams from Japan's Agency for Marine Earth Science and Technology, JAMSTEC, formally identified and named the Mariana snailfish in a 2019 paper published in the journal Zootaxa. The species designation is Pseudolaparis swiri. Lindley's paper described the anatomy in precise terms: a pale, semi-transparent fish with internal organs visible through its skin, evolved over millions of years into a body plan specifically optimized for depths that would instantly kill any conventional vertebrate. The paper is peer-reviewed, institutionally backed, and available in full to anyone who wants to read it. The 2023 depth record was set by an expedition from the University of Western Australia's Deep Sea Research Centre, a group with more than 2 decades of continuous hadal research, using an autonomous lander equipped with ultra-low-light cameras and depth sensors accurate to within 2 meters. 3 independent institutions verified the data before the record was formally acknowledged. Dr. Alan Jameson led the expedition. When he addressed the press after the footage was confirmed, he said the discovery raises more questions than it answers. That sentence, from one of the most experienced hadal researchers on the planet, is the honest starting point for everything that follows. The official public account of the Mariana Trench is clean and simple. It is a geological trench formed by the subduction of the Pacific Plate beneath the Mariana Plate. It has been visited by crewed vessels 4 times: Jacques Picard and Don Walsh in 1960, James Cameron in 2012, and the Limiting Factor submersible team in 2019. Life exists down there, the standard account says, but it is sparse, highly specialized, and barely holding on. The trench is largely mapped and largely understood. That is the official version. What the data shows is considerably more complicated. The prevailing scientific model through most of the 20th century held that no vertebrate fish could survive below 8,000 meters. The biological mechanism behind that limit was well established. Fish cells at extreme depth produce a molecule called trimethylamine oxide, TMAO, to prevent pressure from deforming their proteins. The concentration of TMAO required increases proportionally with depth. Researchers calculated a biochemical ceiling at approximately 8,200 meters. Below that point, TMAO concentrations could not realistically sustain protein function. Below that line, the math said no vertebrate life, period. The consensus was published, peer-reviewed, and taught in university programs for decades. In 2014, a snailfish was filmed at 8,145 meters. The community revised the ceiling and moved on. In 2017, the record reached 8,178 meters. Revision, recalculation, adjustment. Then, in 2023, the University of Western Australia team filmed their fish at 8,336 meters, 136 meters past the revised theoretical ceiling that had already been extended twice. The fish was not struggling. It was not near death. It was moving with the calm, purposeful ease of an animal in its natural environment. The theory was not slightly off. It was wrong by a margin that has not yet produced a satisfying replacement explanation. The depth record is the most vivid single data point from the Mariana Trench in recent years. It is not the most alarming one. In 2019, a study published in AGU's journal Geophysical Research Letters by a team at the Chinese Academy of Sciences in Guangzhou, led by geochemist Ning Wang, analyzed amphipod crustaceans collected from the Mariana Trench and two adjacent trenches in 2017. Amphipods are small, shrimp-like scavengers, numerous, fast-moving, and among the most common large organisms in the hadal-zone. Wang's team analyzed their body tissue for carbon-14, a radioactive isotope. Carbon-14 from nuclear bomb tests has been present in surface ocean water since the late 1950s. More than 500 nuclear detonations between 1945 and 1980 injected carbon-14 into the atmosphere, which was then absorbed by the surface ocean and entered the food chain. Standard oceanographic models predicted that water column circulation would carry surface water to hadal depths on a timescale of hundreds of years. Under that model, Cold War nuclear carbon should not have reached the floor of the Mariana Trench in meaningful concentrations until the 22nd century at the earliest. Wang's team found it in the amphipod tissue at concentrations matching surface ocean levels, collected in 2017, less than 60 years after the bomb tests. The food chain, the constant slow rain of dead organic matter sinking from the surface, had moved the nuclear signature to the deepest place on Earth in decades, not centuries. Wang wrote in the paper, although oceanic circulation takes hundreds of years to bring water containing bomb carbon to the deepest trench, the food chain achieves this much faster. That single sentence dismantles the assumption that the trench is an isolated system insulated from surface world events. It is not isolated. Everything that enters the surface ocean is, through the food chain, eventually delivered to the bottom of it. The timeline is just much shorter than the models said. The mercury data confirmed the same mechanism from a different angle. In 2020, researchers reported that mercury from coal-fired power plants and industrial facilities, primarily in Asia and North America, had been detected in the tissue of animals living in the hadal-zone of the Mariana Trench. The pathway was identical. Surface organisms absorbing mercury during their lifetimes, dying, sinking, and being consumed by trench scavengers. The industrial emissions of the modern surface world were arriving, in measurable concentrations, in creatures living 11 kilometers below it in permanent darkness. The most unexpected single discovery from the NOAA Okeanos Explorer expeditions was not biological. On July 9, 2016, the final operational day of a 3-month expedition, the remotely operated vehicle Deep Discoverer was conducting a seafloor survey near Saipan at a depth of 357 meters. The dive had been planned as a standard site survey following earlier sonar mapping. The ROV cameras swept across the sediment and found something that no mission briefing had prepared the crew to see. A Boeing B-29 Superfortress bomber lay on the sea floor, upside down, largely intact. The B-29 was the largest American bomber of World War II. From 1944 onward, the United States Army Air Forces operated hundreds of them from air bases on Saipan and Tinian, flying long-range missions against Japan. Losses were significant. More than a dozen B-29s went down in the waters around the Mariana Islands and were never recovered. The Okeanos expedition was not searching for them. The broadcast was live. When the wreck appeared on the feed, the scientists narrating the dive were seeing it in real time, on camera, with no opportunity to prepare a framing before the public saw it. The NOAA team confirmed it as the first B-29 crash site from the Mariana campaign ever located. By their own accounting, more than a dozen similar wrecks remain somewhere on the surrounding seafloor, unlocated. If you have never heard any of this before, the fish that lives where vertebrates are biochemically forbidden, the nuclear amphipods, the B-29 found on a live broadcast. Hit subscribe right now, because this is exactly the kind of buried science we cover in depth every week, and you do not want to miss what comes next. The organisms filmed during the Okeanos Explorer dives represent something that NOAA's own scientists, narrating in real time, described as inner space aliens, creatures so structurally different from anything in the shallower ocean that standard biological language felt inadequate. Among the documented finds were new species of hydrothermal vent communities, dense concentrations of life organized around cracks in the seafloor where superheated water emerges at temperatures exceeding 400 degrees Celsius, carrying dissolved sulfur compounds and metals that are toxic to surface life. The organisms around those vents do not use photosynthesis. They use chemosynthesis, extracting energy directly from hydrogen sulfide to build biomass in permanent darkness. They have, in effect, constructed a fully independent ecosystem that runs entirely on chemistry, with no connection to sunlight whatsoever, producing biological output at rates comparable to shallow water reef systems. Hydrothermal vent field on trench floor. White mineral chimneys rising from dark sediment, pale tube worms and shrimp in dense clusters around the vent openings, superheated water shimmering in the ROV lights. The same expedition documented a geological feature inside the Mariana Trench Marine National Monument that had no prior record in United States waters, a petit spot volcano. Petit spot volcanoes form not at tectonic plate boundaries but in the interior of plates, far from any recognized zone of geological activity. They were first identified in Japanese waters in 2006. Their existence challenged the simplified model of plate tectonics that restricted volcanic activity to boundary zones. Finding one inside the monument suggested that the seafloor geology of the trench system is more active and less thoroughly mapped than the standard model described. There is a sound that NOAA and Oregon State University first detected in 2014. Recorded by autonomous underwater gliders conducting an acoustic survey of the trench on the agency's behalf. The sound lasted approximately 3.5 seconds, dropped 2.5 octaves in pitch, and bore no resemblance to any previously cataloged biological call in the marine acoustic database. The research team nicknamed it the biotwang, partly for the twanging metallic quality of its opening tone, partly because no more precise descriptor existed at the time. They spent 2 years studying the recordings before publishing any interpretation. In 2016, Sharon Newkirk and collaborators at Oregon State published their analysis in the Journal of the Acoustical Society of America. Their conclusion: the biotwang was produced by Bride's whales, a species of baleen whale common in tropical Pacific waters, likely using an adapted call to take advantage of the trench's deep-water acoustic properties. The geometry of the trench amplifying and extending the call's range significantly beyond what it would achieve in open water. The source was identified, the sound was explained, at least officially. What was not explained, and what no subsequent paper has addressed directly, is why the call shared no recognizable characteristics with any previously recorded Bride's whale vocalization. The species had been studied acoustically since the 1970s. Its calls were on file, the biotwang matched none of them. The Oregon State researchers proposed that the whale had specifically modified its call to exploit the trench's acoustic environment, but that hypothesis has never been tested in a controlled study. In the published literature, it remains marked as probable rather than confirmed. The sound came from a known species. The specific acoustic behavior that produced it has not been replicated or fully explained. In February 2026, a month-long expedition to the Mariana Trench, using autonomous submersibles operating between 8,000 and 11,000 meters, reportedly identified 3 previously unrecorded species. According to available reporting from marine biology research outlets, the finds include a translucent fish with bioluminescent organs in a configuration not previously documented, a new amphipod species, and a colonial organism, a creature that exists not as a single individual, but as a coordinated community of genetically identical units acting together, of a type with no close analog in any previously cataloged hadal fauna. The colonial organism, according to the expedition scientists' descriptions, reportedly produced bioluminescent pulses between its individual units in a pattern they described as structured. ROV footage of a long, faintly luminous thread-like colonial organism drifting in black water, sequential blue-green light pulses moving along its length in a repeating, non-random pattern. That word, structured, is the precise point of interest. Bioluminescence in deep-sea animals has been documented for decades. Creatures flash and pulse for understood purposes, attracting prey, startling predators, signaling mates. These are reflex-driven stimulus-response behaviors. What structured suggests, in the context of inter-unit coordination within a colonial organism, is something beyond reflex, patterned, repeating, and variable in ways that do not match simple mechanical triggering. No peer-reviewed paper describing this organism had been published at the time of this recording. The exact details of its behavior remain under analysis. What is confirmed is that it exists, that it was filmed at hadal depths, and that the scientists observing it in real time did not have an immediate category to put it in. The deep sea covers approximately 65% of the Earth's surface. The hadal zone, everything below 6,000 meters, has been surveyed in detail across less than 1% of its total area. The 4 crewed descents to Challenger Deep produced a combined observation window of perhaps 6 to 8 hours at full depth. The scientific community's current working estimate is that between 1/3 and 1/2 of all hadal species remain undiscovered. That figure does not come from advocacy. It comes from the rate of new species identification per unit of sampling effort, tracked systematically across 30 years of hadal research. Every improvement in the instruments produces more new organisms, not fewer. The trench is not approaching a complete inventory. The inventory is growing faster as the tools improve. Now factor in the contamination data. Ning Wang's 2019 conclusion that the deep ocean is not an isolated system was not a rhetorical statement. It was a quantitative finding from a peer-reviewed study. The food chain delivers surface contaminants to the hadal zone on a timescale decades shorter than physical ocean circulation would allow. Cold War nuclear carbon is in the amphipods. Industrial mercury is in the fish. The surface world's chemical history is being archived in real time in the tissue of organisms living at the bottom of the Pacific Ocean, and the archive is updated continuously by the constant downward drift of the food chain. Visual note: wide-angle seafloor shot of a whale fall, a massive skeleton resting on trench sediment, surrounded by dense clusters of pale hagfish and smaller organisms, bacterial mats glowing faint green across the bones. When a whale dies in the open ocean, the carcass sinks and becomes a whale fall, an event that can sustain a complex community of scavengers and chemosynthetic bacteria for 50 to 100 years. The hagfish arrive first and strip the soft tissue. Sleeper sharks and crustaceans follow. Eventually, the bones themselves are colonized by sulfur-eating bacteria that extract energy from the lipids in whale bone marrow. At any given time, dozens of whale falls in various stages of decomposition are presumably occurring across the length of the Mariana Trench, each one a temporary hub of biological activity on the sediment plain. The ecosystem down there is not sparse and barely surviving. It is fed continuously from above, sustained by processes that science spent most of the 20th century insisting were too slow and too dilute to matter at those depths. Lay all of it side by side. A fish living 136 meters past the confirmed biochemical ceiling for vertebrate life. Nuclear fallout from 1950s bomb tests circulating in the tissue of trench animals 60 years later. Mercury from surface world power plants accumulating in hadal fauna 11 kilometers below its source. A World War II B-29 discovered intact on the seafloor during a live broadcast government dive, the first of more than a dozen unlocated wrecks from the Mariana campaign. A sound that took 2 years to identify because it matched no known vocalization from its source species. A colonial organism filmed in February 2026 exhibiting bioluminescent behavior that the observing scientists described as structured and, reportedly, variable in ways that exceeded simple reflex. And beneath all of it, a geological environment generating petty spot volcanoes inside tectonic plates and supporting chemosynthetic ecosystems that run on hydrogen sulfide in permanent darkness. Final wide ROV shot from trench floor looking upward through thousands of meters of black water, a barely perceptible trace of surface light impossibly far above, the weight of the ocean visible in the darkness between. None of this needs a supernatural explanation to be extraordinary. None of it requires a conspiracy to be disturbing. It requires only an honest accounting of what the data shows versus what the standard model predicted. And the gap between those two things is wider and more consistently one-directional than any official summary of the Mariana Trench has ever acknowledged in public. The model said the trench was sparse. The footage shows density. The model said the ceiling for vertebrate life was 8,200 meters. The footage shows 8,336 meters and counting. The model said contamination from the surface took centuries to reach the bottom. The tissue analysis shows it takes decades. The researchers at the University of Western Australia, at NOAA, at JAMSTEC, and at the Chinese Academy of Sciences are not claiming that the trench contains monsters. They are claiming something considerably more specific and considerably harder to dismiss, that the place is less understood than the public account suggests, that the rate of genuinely new discoveries is accelerating rather than decelerating, and that the theoretical frameworks used to describe the trench were systematically wrong in the same direction, toward underestimating what is down there. Every single time they were tested against actual footage. The fish at 8,336 meters is the clearest example of that pattern. Every time the cameras improve enough to look, the cameras find something that was not supposed to be there. The one question that the current body of evidence does not answer, and that keeps the researchers going back with better instruments year after year, is this: if the food chain delivers the surface world's history to the hadal-zone faster than any physical model predicted, and the biological diversity down there is substantially greater than the public account described, then what specific organisms are processing that input at depths we have never filmed, in areas of the trench that represent more than 99% of its total floor area, and organizing into behaviors that existing science does not yet have the vocabulary to classify? Drop the single discovery from this video that you think the public should know more about in the comments, and tell me why you chose it. That conversation is worth having